Welcome to another of the eBiology Teacher Podcasts. I'm Mr. Doc, and today we're going to be going over protein synthesis. This is the third in a three-part series on understanding how DNA and RNA play a role in making sure that we are able to live. All right, because without them we can't. Today we're going to talk more in depth about the function of mRNA, rRNA, and tRNA, which we were introduced to in the first of these series. We're going to talk about two processes in protein synthesis that are important, that is transcription and translation. We're going to talk about why protein synthesis is important at all, because why am I learning this kind of thing? And then, of course, why is being able to translate and transcribe DNA into amino acid, how do we do that? Can we speak the language of DNA? So let's go ahead and get started. What we're going to do today is start with the idea about the code. Okay, we've been told that DNA is the code. Well, that's true. You see, DNA can be converted and can be translated into protein. Proteins are important because they make up a lot of things that cells need. For example, enzymes. If you remember in DNA replication, we said that DNA has to be able to replicate in order for things to pass on the hereditary information to their offspring. Well, yeah. How are we going to do that if we don't have enzymes? Because polymerase and helicase have to be there for us to do that. Enzymes are proteins. So proteins are incredibly important, if not essential for life, and anything that's essential for the life of a cell is certainly essential for the life of an organism. Now, there are three types of DNA that we're concerned with. The first is mRNA. mRNA transcribes information. That's what it does. It gets information from the DNA inside the nucleus, which is where the DNA is, and then it takes it out to the cytoplasm where ribosomes are able to translate it into amino acids using tRNA. Now, this seems like a lot to think about right now, but the process is pretty simple if you know about complementary base pairs, and that's something we spent quite a bit of time on. Now, to talk about complementary base pairs, what we need to do is look at the labs that we've been doing in class. Guanine pairs with cytosine just like cytosine pairs with guanine. Adenine pairs with thymine just as thymine pairs with adenine. The only difference in RNA is that there is no thymine in RNA. There is only uracil. So anywhere you would expect to see a thymine, there'll be a uracil in its place. All right. Now, one thing we do want to mention real quick is that DNA is a double strand, RNA is a single strand, whatever. We'll go over those similarities and differences in just a little bit. But transcription works like this. And this is the first step to protein synthesis. The first step is to transcribe the information. In other words, what happens is RNA polymerase, because DNA polymerase works in replication. DNA polymerase is involved in DNA replication. RNA polymerase is in protein synthesis. Sorry, RNA polymerase unzips the DNA molecule like you see here where the little hand is. It's unzipping it. Then what happens is mRNA is formed by complementary base pairing. But instead of the nucleotides looking like this with a deoxyribose sugar, they're going to look like this and have a ribose sugar, which is a 5-sided sugar. And of course, if it's got a different shape, we know it's got a different function. Exactly. So the deoxyribose sugars are part of the DNA chain. And what will happen if we were looking at this model is going to split open and RNA nucleotides are going to start to fit in there. Then what happens is we end up producing a RNA strand, and that's why it's important to understand that RNA is single-stranded and DNA is double-stranded or double helixed. You'll notice that the DNA is not affected by the creation of mRNA. mRNA is created off to the side. DNA opens up, gives away its information, closes back up, and it remains the same. Okay? Now, actually, I'm going to go back here and see if I can't launch this animation here. And we're going to open this little animation, and I'm going to try to show you what's going on in terms of this little animation here, okay? So hold on oh, here it comes, here it comes. Alrighty, 1 second. I know it's hard to see, so let me get some things under control here. Okay, now this is transcription. What happens here is the RNA polymerase is going to come up and it's going to attach to the DNA. Now there's a thing called the promoter, and that's where RNA polymerase knows this is where I'm going to start copying. What happens is the RNA polymerase starts to unzip and everything starts to move across. And then we start to add those nucleotides, boop, 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 boop. And then we're going to hit what's called a terminator. The terminator says, okay, stop copying here. So we have a promoter that says start and a terminator that says stop. Then the polymerase falls off. Boom, we have a piece of mRNA. This is produced through the process called transcription. Okay? Now, let's talk about the next step. But first, now that we have RNA, don't we really need to talk about what's the difference? tRNA, listen to me. I'm having a rough day today, I apologize. DNA has a deoxyribose sugar, which is 6-sided, as you see on the little diagrams. RNA has a 5-sided sugar that's called ribose. So DNA has deoxyribose, RNA has ribose, which is a 5-sided sugar, RNA, DNA. DNA contains guanine, cytosine, thymine, and adenine, whereas RNA contains guanine, cytosine, adenine, and uracil. And the pairs are slightly different. In DNA, A pairs with T, T pairs with A, C pairs with G, G pairs with C. But in RNA, A pairs with U, U pairs with A, and then of course C and G remain the same. That means that in RNA, anywhere you would expect to see a T, there's going to be a U in its place. All right? Now, let's go to talk about language. Okay, remember we talk about this being a language all the time. That's kind of the process we've been doing. Well, if we have words in the English language, DNA has words called codons, and they are a series of 3 nucleotides that mean something. As a matter of fact, they mean a specific amino acid. So every 3 nucleotides on a strand of mRNA is going to code for a specific amino acid, and a series of amino acids forms a polypeptide chain, and that polypeptide chain is called a protein, and that's where protein synthesis comes in. So if you can read every 3 letters on an mRNA chain, and of course mRNA being translated from DNA, then you can determine what proteins are going to be in place. And the way we do that is using what's called a codon chart. Now, the codon chart, which I have on the screen twice here because we're going to have to change the picture in a minute and I want to be able to use this thing, is a way to figure out what protein we need or what amino acid we get based on a specific codon. Hold on 1 second. Let me find my pencil here. Alrighty. Now, let's say that we have a codon. Now codons are on mRNA, right? Let's say we have a codon, that's 3 letters, and the letters are CUC. In other words, in that order, we have cytosine, then we have uracil, then we have cytosine again. Here's how we do this. The first base we locate on the left-hand side of our chart, C. The next one we locate on the top of our chart, U. And then the last one we locate on the right side of our chart. What did I say? CUC? That's right. All right. So we found CUC. Trace it back. Here it is on my chart. It's leucine. That is the amino acid that's going to go into place. Let's do that one more time just so everybody's kind of comfortable with it. All right. Let's say we run into an amino acid where it's CCA. CCA. C is my first, the C is my second, so I know that it's going to be in this box right here, and then I find A, CCA is right here, right? So the next amino acid is going to be proline. That's how I use the codon chart. Now there is another way to do this, and we're going to have these in class, but this is called the codon wheel, and basically what we do is we line up the letters, so A, G, C would be serine, and so forth. We'll learn how to use this in class. Alrighty. So let's talk about the process. First off, tRNA is going to be involved. okay? tRNA is going to be important because tRNA is what brings the amino acids to the party. They are the designated driver. The thing about tRNA is that tRNA has to match the codon with what's called an anticodon, and it's very simple. Try to imagine if we have a codon, and the codon is GCC, as you see right here in the diagram. GCC. That's the codon, okay, that codes for amino acid. The anticodon on tRNA just has to be the opposite based on the complementary base pairs. So GCC is the codon, then CGG has to be the anticodon because GCC, the opposite of G is C, the opposite of C is G, the opposite of C is G. GCC, CGG. Okay? Now that means that every tRNA that has CGG as its anticodon has this amino acid. Well, the question is, what is that amino acid and how do I know I need it? Well, let's take a look back to our codon chart. The codon is actually GCC. So we go to our chart, right? We say G is our first one. C is our second one, so we know we're dealing with it here. C, GCC is the codon, so alanine is the amino acid. That's how that works, okay? Now, let's continue on a little bit, and we can see here that mRNA travels through the cytoplasm, okay? It's the list of instructions. tRNA comes to the ribosome bringing the amino acids. mRNA has what's called a start codon, and the start codon is always AUG, okay? The reason this is important is because the start codon tells us that we can start building proteins now, and I'm going to highlight that in blue instead of red because that could be confusing. Every protein starts with the same amino acid because its codon is the start codon, and remember, a codon pairs with a single amino acid. So methionine is the first amino acid on the block when we're talking about building proteins. So AUG, when we're talking about protein synthesis, AUG is the green light. It's what tells us to start. Now, this process is going to continue, and the different amino acids are going to be continued to added until we reach a stop sign or a stop codon. The stop codon tells the ribosome, hey, you're done, wrap it up and let's go. Once that's done, the protein can be used for whatever it's supposed to be done. The stop codons are not quite as simple because there's not just one of them, there's three of them. And if you look at your codon chart, we can find that UAA, UAG, and where's the other one on our chart? Here it is, UGA. These are all stop codons, and that says, okay, we're not going to add any more amino acids here. We're simply just going to wrap it up, okay? Now, I am going to show you another animation that is similar to the one that I just showed you, all right? There we go. And this one is about translation, obviously. So now we have a piece of mRNA. Hopefully you can recognize that this is mRNA. The next thing that's going to happen is the ribosome is going to fit onto the mRNA. And remember that mRNA, or a ribosome, has two pieces. It's got a little small piece, or a small subunit, and then a large subunit. The tRNA is going to attach as well. Now, I want you to notice on the mRNA, this codon, AUG. AUG is the start codon, and it codes for an amino acid called methionine, and there's methionine. Now, if we continue, the ribosome clamps down, and we continue to copy. Another tRNA is going to come in here. If we go AAG, that's our next codon, we look at our chart, and we say AAG, lysine should be our answer, and if you look right here, this is lysine, LYS. Continuing on, the methionine is going to attach. So what happened is the methionine went from here, boop, to there. Next thing that's going to happen, we're going to move on. The old tRNA is gone. The new tRNA is going to come in, and it's going to land down. Now notice that the codon here is UUU. The anticodon is AAA because they have to fit, right? The opposite of U is A. So let's look at U, U, U. phenylalanine. Yeah, I'm not really concerned that you know how to pronounce them. I'm just concerned that you are able to notice that these are the amino acids that are coming in. Protein moves over. We slide. The old tRNA goes. The new tRNA comes in. GGC is the codon. This one should be GGC glycine. And if you look closely, GLY, there she is. We continue. Now, we go to our next one, and it's UAA. Well, UAA is the stop codon. So if we've done this correctly, the next thing that should pop into place is what's called an RF, or a release factor. So we bring in the release factor instead of a new amino acid, and then when we're done, boom, we have a protein, we have the release factor, we have all of the tRNA, and then the ribosome just breaks apart and we're done. And that's how we make proteins. So you could spend some extra time, and what I suggest you do is look at this page and sort of ignore the tRNA and the protein and see if you can translate the mRNA to tRNA and then the proteins, keeping in mind, by the way, that to find out a codon, it's on the mRNA. So let's go ahead quickly and look at our final objectives. First of all, describe the function of mRNA, RNA, rRNA, and tRNA. mRNA is the messenger RNA. It copies the information or transcribes the information from the DNA in the nucleus and takes it out to the cell. rRNA is what makes up the ribosome where it can be read, and tRNA brings in the amino acids. Transcription is the part where mRNA is made. It's copying information from DNA. Translation is where that's literally being translated into a series of amino acids or protein. Why is this important? Well, you need proteins. You need enzymes, and you need proteins for muscle fibers and fingernails and hair and all that other stuff. So it's incredibly important. And the question is, can we transcribe and translate DNA sequences? If you can look at a piece of mRNA and divide it into series of 3, starting with AUG, the start codon, every 3 letters is a new amino acid. So you should be able to build a chain, and if you run into UGA, UAA, or UAG, and that's 3-letter series, that's the stop codon, which means, boom, you're done. All right. I hope this has been helpful. I know this is sort of a tough subject, so if you want to, go back and replay this again. Hopefully it'll help you. Or, of course, if you have questions, you can always come in early in the morning or maybe after class or something and talk to me about it. I'll be glad to help. So there you go, protein synthesis. We'll finish this up, and then our next set of notes is going to be on mitosis and then meiosis, and then we'll wrap this whole cellular reproduction up. Saved by the bell.